Yeah, so today I'll be presenting this paper that we just published last week in Nature Genetics. Yeah, so we basically looked at, de novo variants in the context of the exome aggregation consortium's data set. So first off, most of this paper deals with, autism spectrum disorders, or ASD. And just so we're all on the same page, ASD is defined in the DSM-5 by impairments in two core domains. It's communication and social interaction, and restricted interest and repetitive behaviors. It has a prevalence in the US of about 1 to 2%. Worldwide, it's about 1%. There's a male bias in that there's 4 males for every 1 female with ASD, and it's a highly genetic heritable disorder with heritability estimates, upwards of 90%. And also feel free to stop me and ask questions at any time. So over the past 5 years, basically the main success in unraveling the genetic etiology of ASD has come from studying de novo mutations. And de novo mutations are those mutations that arose spontaneously in the egg or the sperm and are inherited are present in the child but not in the parents. And so when we examine when we look at the rate of these de novo mutations in children with autism and their unaffected siblings, we see an enrichment of both de novo missense and de novo protein truncating variants in children with autism against expectation, while the rate of these de novo variants is the same from what we would expect by chance for the unaffected siblings. And so these initial analyses then identified, both de novo missense and de novo protein truncating variants as a class of variation that's then associated with autism. Beyond just a excess of these mutations compared to expectation, there's a number of other, consistent findings across these studies that have basically been done over these past 5 years. The first is that there's a higher rate of both de novo protein truncating variants and, to a lesser extent, de novo missense variants in children with ASD compared to their unaffected siblings. There's a higher rate of de novo missense and de novo protein truncating variants in females as compared to their male counterparts. And this is consistent with this idea of a female protective effect in that, there's a bias towards more males with autism, so it seems like you need a greater burden of mutations for a female to get diagnosed, and so that's why we see more of these mutations in females. And so this is kind of lends some credence to this hypothesis. Whether it's diagnostic or biological is another story. And lastly, that the rate of de novo protein truncating variants is higher in children with autism and intellectual disability than those with just autism by itself. And so when we basically collect all the de novo variants that have been published in children with autism over these past 5 years, we basically have close to 4,000 children with autism and a little bit more than 2,000 are unaffected siblings. And this is work in collecting these variants and standardizing their annotation. That was done by a former graduate student in the lab, Caitlin Samoa, and Stefan Sanders from UCSF. And so as a graduate student, I was reading all these papers. This is basically what the field of autism genetics has basically evolved into to some degree. And you'll notice that there's a number of these very consistent implicit assumptions that's made about de novo variants, namely that they're rare, they're going to be highly penetrant, and they're unlikely to be observed in other individuals. So if you see one of these de novo variants in a gene that's considered to be deleterious, then it's assumed that it causes the disease, and you're not going to see this de novo variant in anyone else. But we know that mutation is not uniform across the genomes. There's certain portions that have a higher mutation rate than others. And so I kind of set about questioning these assumptions. And we started off with this very simple question of how often do we see one of these de novo variants in one of our children with autism that's present in basically other individuals in the population? So here we have, a classic trio, your mom, dad, and child, and the child has a de novo variant, and I was just wondering, does anyone else in the world have one of these de novo variants? And so, of course, we needed some population and some cohort to represent our general population. So luckily, the Exome Aggregation Consortium was basically just arrived at this point, and this was put together by Daly, MacArthur, and Monk-Leck, and this was a reference exome database that's an order of magnitude larger than what was previously available with the NHLBI-ESP data set, and then it's, of course, 60 times larger than 1,000 genomes. So this is just a ton of individuals, none of which have autism. And while it's not a perfect representation of the ethnicities of the world, it's certainly a lot better than what we had previous to this. So when we actually went in to EXAC and we asked whether any of the de novo variants that have been seen in children with autism or unaffected siblings or in any of the individuals in EXAC, we arrived at this surprising finding that basically a third of these de novo variants in both individuals with autism and their unaffected siblings are actually present in EXAC. So these are sites in the genome where we observed the mutation occur, and these mutations have occurred prior in human history. So this was surprising because, again, there was this, there's this assumption that these are rare, they're highly penetrant, and they shouldn't be seen in other people. So the fact that a third of these, are seen in other individuals is, striking. And basically, when we saw this, we basically had to ask, is this real? And for the most part, a bulk of this paper is basically trying to prove to ourselves and to the readers that indeed these are real de novo variants, and and they're not just false positives. So one of the first things we did is we wanted to see, well, if we're observing sites in the genome that have undergone multiple mutations, they should probably be at sites that are more likely to mutate. And so indeed, 71% of these de novo variants that we saw in someone in EXAC is at one of these CPG sites. And CPG sites have basically a mutation rate that's an order of magnitude higher than the rest of the genome. So it makes sense. It gives a biological basis for what we're observing. So that was very reassuring. This was really good. We also checked to see if the allele frequency of these de novo variants is the same across both, intellectual disability, autism, and their unaffected siblings, as well as comparing that proportion to EXAC. And you'll see, across each of these different allele frequency thresholds, there's no difference in both the rate across these different cohorts and in that proportion to EXAC. And this is a comparison we're going to do, you'll see throughout this paper, is comparing rates of de novo variants across, intellectual disability, autism, and unaffected siblings. And so again, this is also reassuring that, we're not seeing anything wildly different. And there was a number of other analyses that we did. We wanted to make sure that these de novo variants in EXAC and not in EXAC are associated with paternal age. That's been a consistent finding. They were. There's no difference in the rate of de novo variants in and not in EXAC depending on which study we took these published de novo variants from, whether they were validated or not validated by Sanger sequencing. So all of these checks were coming out correctly. So basically, now that we had convinced ourselves that this is indeed real and not just a bunch of false positives, we can now ask whether the de novo variants in EXAC are any different from the de novo variants not in EXAC, or do we just make some random cut and that's about it. So the first thing we did was we wanted to check to see what the rate of de novo synonymous variants were. And so here what I've shown you is the rate of all de novo synonymous variants, and you'll see that there's no difference between the rate in intellectual disability developmental delay in blue, autism in purple, and the unaffected ASD siblings in green. And so when you don't see any difference in the rate of some class of variation across your different cohorts, then that means that it's not associated, and indeed no one else has seen an association for synonymous variants. So that's good. So when we take a look at just those de novo synonymous variants in EXACT, we don't see any difference in the rate across the different cohorts. So this is consistent with a a class of variation that is not associated with your case status, and so this is good. Then when we turn to our de novo protein truncating variants, and these were the variants that showed the greatest the greatest enrichment autism and intellectual disability, we see if we look at all de novo protein truncating variants, there's a twofold excess in intellectual disability and developmental delay, and about a 1.6-fold excess in autism. Now we do the same thing. We look at just those de novo protein truncating variants in EXACT. Now we've completely obliterated this, very strong signal. And in fact, you'll notice that there's actually a higher rate in unaffected siblings. So unaffected siblings have a higher rate of de novo PTVs in EXACT. So mathematically, when you remove those, you see a greater enrichment than what you saw before for those de novo protein truncating variants, not in EXACT. And so, this is good because basically we can throw a third of our data away and we get the same association. And in fact, not only is it the same, but it's actually stronger than what we've had before. So this is basically the fact that these de novo variants that are present in EXACT, we don't see any difference between their rate in cases and controls, and so this is consistent with the class variation that's largely benign. And so in the words of Obi-Wan Kenobi from Star Wars, these aren't the de novo variants you're looking for. So all of these analyses that I've discussed so far have been focused on at the variant level, whether a given variant is present or absent in EXACT, whether that variant is more or less likely to be associated with disease. And so now we can move up to the gene level and ask whether these de novo variants that are absent in EXACT reside in genes that are predicted to be loss of function tolerant. And so we use this metric by Caitlin Samoa called PLI, which is just the probability of loss of function tolerance. And indeed, when we look at these genes that are more likely to be loss of function tolerant, we see a much higher excess. So when we take a look at where we were, we started at twofold excess in intellectual disability and 1.6-fold enrichment in ASD, and we removed those de novo protein truncating variants in EXACT, and we removed the de novo protein truncating variants that are not in EXACT, but were in one of these genes that tolerates loss of function mutation. Then we arrive at this massive enrichment with like a 6.7-fold excess in intellectual disability and a more than a 3-fold excess in ASD. And so this is we basically doubled or tripled our effect size just by removing these variants that are just present in other people or in these genes that don't are not suspected or tolerate these protein truncating variants. So one thing, that we were concerned about is the fact that EXACT does have, there's a lot of exomes in EXACT that are from schizophrenia cohorts. And so does this bias our results? And in fact, if you just restrict to the version of EXACT that has all the psychiatric exomes removed, you actually really you can barely even tell the difference. The effect size is a little bit smaller, but that's because you threw away 15,000 exomes. So whether you're using the full version of EXACT or the schizophrenia, the non-psych version of EXACT, at least for intellectual disability and autism, it really doesn't make any difference. And so, for our analyses, it definitely made sense to have another 15,000 exomes from which we can basically just remove these variants that were just are less likely to confer risk to disease. So if you remember, there was a bunch of these hallmarks of autism de novo studies. One is this male to female ratio, and the other is the skew in the IQ distribution. So if indeed these de novo variants that are absent from EXACT are, real and confer more risk, then we should see the same enrichments and patterns that we saw with the full data set. So when we look at that male to female ratio, and here what I have on the far left side is the de novo variants that are not in exec and in one of these loss of function constrained genes. You'll see here that the male to female ratio is about 2 to 1. So there's 2 females for every 1 male, and this is 6% of our cohort. If you look at all the other de novo protein truncating variants and all the other individuals that don't have a de novo protein truncating variant, here that male to female ratio is like 1, and their IQ is the same. So we don't even see much of a difference, at least with regard to like IQ and that male to female ratio between individuals that don't have a de novo protein truncating variant and individuals with one of these de novo protein truncating variants that is either in someone else or in one of these genes that is not, that tolerates such mutations. And then when we look at that IQ, the IQ distribution, we see that the de novo protein truncating variants that are not in exec still see that same skewing, that children with ASD and intellectual disability have a higher rate of de novo protein truncating variants not in exec, and that those de novo protein truncating variants in exec, in fact, are like uniform across this IQ distribution. So these variants are clearly not playing a role in their IQ, which is reassuring. Now, if we want to start looking at these rates and split by sex and IQ, here what I've done is we have intellectual disability and developmental delay, and then autism is now split. The dark purple is ASD with ID, and then the light purple is ASD without ID. And so you can see that the individuals with autism and intellectual disability have a much higher rate of de novo PTVs than the individuals with just autism, which is basically what we saw before. And then if we split by sex here, we can see that intellectual females have a higher rate of de novo PTVs not in exec in one of these loss of function constrained genes than their male counterparts. Then for ASD, you see the same pattern, that basically females have a higher rate than males, and individuals with ASD and intellectual disability have a higher rate than just ASD alone, though these associations, because we're just cutting our data into like smaller and smaller chunks, the error bars get large. And so if you compare the differences between each, between both females and males, or females with intellectual disability and females without intellectual disability, it's not significant. But you can see that these, there's this nice staircase pattern. And then all the other de novo variants, we don't see any association there. The other thing we were wondering is like if this analysis framework, in that if we remove variants that are present in exec, and if we remove variants that are in these genes that tolerate loss of function mutations, if we can find some excess in inherited protein truncating variants. And so indeed we actually do. So in the top box, what I have is, you can see that there's that ASD association for de novo protein truncating variants, and there's no association for all the other de novo protein truncating variants. But then if you look at the inherited variants on the bottom, you'll see that those inherited variants that are not in exec and in one of these loss of function constrained genes, we have a modest increase over the de novo protein truncating variants that were not inherited. And so you get an odds ratio of about 2. And then there's another 14,000 of these protein truncating variants that are either in exec or in one of these genes that tolerates loss of function mutations. And you see no association between whether they were inherited or not. And so this is interesting because there's just a very small fraction of these protein truncating variants that are inherited that actually show association versus outside of this, massive, excess. So it's a really small fraction, and clearly, we need to do a better job of trying to figure out which of these inherited variants actually confer risk or not. But this was a step in the right direction. And lastly, if we kind of like look at the genes that show the greatest excess for de novo protein truncating variants, you'll see that, you know, basically all those genes that were significant are still significant, and they all have these high PLI values. Interestingly, most of these genes show basically have like no inherited variants. It's very, very rare that we see an inherited variant. There's a few instances of these variants in cases. But what I think was most striking was that there's actually a higher rate of de novo protein truncating variants in intellectual disability and developmental delay in like ARID1B and SYNGAP1 and, you know, ADNP and SCN2A than ASD, which might suggest, and we're kind of following this up, that these genes actually confer more risk to intellectual disability and developmental delay than autism. Of course, you know, depending on the diagnosis. I mean, there's a lot of caveats with that statement, but it's a rather striking finding that, you know, that we're seeing a much far more of these variants in different phenotypes. So in summary, you know, basically all of the excess for de novo and inherited protein truncating variant signal resides in these variants that are absent in EXAC and in genes that are intolerant of truncating mutations. And basically all individuals with ASD carry about 3.5 rare PTVs that are de novo or inherited, but only 4% of these, you know, actually occur in one of these genes that's intolerant of such variants. Yeah. And so with that, that's basically the end. And so if anyone has any questions, be happy to answer them.
Thank you, Jack. You can stop sharing the screen now.
Okay.
It was a very cool presentation, I have to say. Not that I'm surprised. You can also, if you want, you can put on your screen. One question I have: did you did the same analysis again, including like a Misson Z-score and NOMAD data? Would you expect that the things improve?
We know that, like, we've done, because the NOMAD VCF is still not
I know.
completely finished. We haven't done a whole lot with that.
But would you expect that's improving?
Yeah. So we looked at just so far we've kind of only looked at the recurrence rate, and we see that it's gone up from 1 in 3 to now 2 in 5. So it's about 40% of these. We'll obviously need to do like to redo all these checks, and the other